Hello everyone! Today we are going to work on learning about cellular respiration. So I know you've already went over this in a good bit of detail in your lesson module so far, but I find some students really find it beneficial when people take the time to just draw out each step-by-step process within this grander process of cellular respiration. So just a warning, this is going to be definitely one of the longer videos that you're going to find in your module. However, a lot of students have said that going through this step-by-step is helpful. So let's get into it, shall we? A couple of things that I want to clarify before I start drawing stuff out. I'm going to be marking all of my enzymes in the color green. So green are our enzymes. Remember, enzymes are things that catalyze chemical reactions. And I'm going to be using our different substrates in the blue color. So we're going to be starting with glucose is where we're going to start, but as we go through the process, you'll see that glucose quickly gets converted into a litany of other things through different chemical processes to help us end up producing energy. And then we have arrows throughout, right, to indicate movement in our chemical reaction series. Now, something that a lot of people forget to go over are the differences between these two arrows, and they actually mean different things. If you see an arrow that is unidirectional, that means that the reaction that's taking place cannot go in reverse. It can only go in one direction. However, if you see a double-sided arrow, one that can go back and forth, these are what we call equilibrium reactions. And so these can go back or forwards, and it's all dependent on the concentration of the substrate, all right? And so I'll give you an example as we go through this. Let's start from the top with cellular respiration. You, as an example, you ate food, and in your food is glucose, and your body is now going to use that glucose that you ate to produce energy. So as like a brief recap, where's one of the sources that you can get glucose? Down the line, either directly from plant matter because you ate plants, or you ate something that ate plants, right? So photosynthesis is a large contributor to us providing us with glucose, right? So we're starting out with our glucose molecule here, and we're starting out in the bloodstream. So this is after, digestion and stuff has taken place, and your food particles have been broken down into their small subunits until we are left with little glucose molecules, all right? So in your bloodstream, we have glucose molecules, and the glucose is going to enter into a cell, leaving your bloodstream and entering into a cell. So this is my plasma membrane of a cell. So here we're in the extracellular space, right? Because this is outside of the cell. Inside of the cell in here, this would be our cytosol. So let's go ahead and label when the glucose enters, it enters into the cytosol of the cell initially. And it enters via a receptor. And this receptor in humans is triggered by insulin, right? So when insulin binds to its receptor, it triggers a response that ultimately allows glucose to enter the cell. Boop. So now we have glucose in the cytosol of our cell. So insulin triggers glucose transporters. Sound good? So here's our glucose, little diamond guy in the cell, in the cytosol of the cell. And now that glucose is in the cell, the first step that happens in cellular respiration, once we're inside the cell, is glycolysis. So remember, glycolysis comes in two steps. We have a step that requires energy to convert our glucose molecule into something else. And then the second phase is the energy producing phase, where we then gain energy moving through our series of steps. Sound good? So step number one, we have our energy requiring phase. So that means that ATP is being used up for this phase. So the first thing that happens is a reaction that is carried out by a kinase. It's hexokinase if it's outside of the liver, or glucokinase if we're looking at a liver cell. You do not have to know that level of specificity, just know that it is a type of kinase, okay? The specific names I'm not gonna harp you on, as long as you know the kind of enzyme is a kinase. And this is important for you to know because all kinases are a byproduct of the conversion of ATP to ADP. Or the reverse of this. Really what a kinase does is it moves phosphate groups around, and this usually either produces or uses up energy, okay? So here, basically what happens is the kinase will take one of the phosphates. Remember, adenosine triphosphate, so we have an A and then 3 P's attached, and that's an ATP molecule, right? So basically what a kinase does is it takes one of those phosphates and tacks it on to whatever chemical it's reacting with, all right? So now we have something called glucose 6-phosphate, and that cost us energy, making that glucose 6-phosphate. And the reason that it's called glucose 6-phosphate is not because it has 6 phosphate groups, it's called that because that is the carbon that the phosphate is attached to. It's on the 6th carbon of the glucose molecule. Sound good? And for any of you who like to go above and beyond, and you have heard about the term glycogen. Remember, glycogen is something we talked about way back when in Unit 1. Glycogen is how animals store their energy, right? That's how they can store glucose to be used later, right? This is where we get glycogen from, actually, is a side reaction can happen here if there's a lot of glucose 6-phosphate building up in the cell. Through a different process, through different enzymes, glucose 6-phosphate can be converted into and stored as glycogen. Sound good? Okay, so that's that. That's just step one now of the energy requiring phase. So let's keep moving forward. Once we have glucose 6-phosphate, we have an equilibrium reaction. So we're going to break down what that means in a second. And that equilibrium reaction is carried out by an enzyme called isomerase. again, we can tell that this is an enzyme because it ends in the word ase. And I will not require that you memorize that name, all right? The ones that I really want you to memorize are the ones that, what kind of enzyme we're dealing with, all right? If there's a specific type, such as a kinase. Sound good? So, isomerase will take glucose 6-phosphate and basically convert it into fructose 6-phosphate. Basically, it rearranges glucose 6-phosphate. Sound good? So, the key thing to note here is this nice double-sided arrow, all right? That double-sided arrow tells us that we are dealing with an equilibrium reaction, which means that the concentration of glucose 6-phosphate, if it's higher than the concentration of fructose 6-phosphate in the cell, then a cell will use isomerase to convert glucose 6-phosphate into fructose 6-phosphate, all right? But if the cell were to have a higher concentration of fructose 6-phosphate, then isomerase would actually do the reverse of this, and it would convert fructose 6-phosphate back into glucose 6-phosphate, all right? So the increase of glucose 6-phosphate is what drives this reaction forward, okay? So we need that increase in glucose 6-phosphate to drive the reaction forward, moving further down into cellular respiration to produce energy. So next, now that we have our fructose 6-phosphate, we have another enzyme coming in, and this is called phosphofructokinase. Okay, phospho relating to the phosphate group, fructo referring to the fructose aspect, right? And then kinase. Kinase is the big thing, right? So we're dealing with something that is going to require ATP. So we're going to be using up ATP again. So that way it can steal one of those phosphates and tack it on. So now our ATP is ADP. It's going to take one of those phosphates from ATP and tack it onto our fructose 6-phosphate to make fructose 1,6-phosphate. All right? Again, the 1 and the 6 are just referring to where in the molecule we will find those phosphate groups. And I apologize, I misspoke. It's actually fructose 1,6-bisphosphate, right? Because there's 2 phosphates. So the Bi is because there's 2 phosphate groups. Sound good? So fructose 1,6-bisphosphate. Here's where things get kind of crazy. So at this point, before moving forward too much, let's zoom out a bit and assess what we've done so far, all right? You ate food. The food was broken down into glucose molecules. Well, the carbohydrates were at least, right? Broken down into glucose molecules. Insulin helped allow that glucose to enter the cytosol of your cell, where glycolysis is able to take place. Just the first part of glycolysis. We haven't done the second part yet. And so at this point, we have used up 2 ATPs, right? 1 ATP was used up here, 1 ATP was used up there. So now we're moving on to the second phase of glycolysis, which is our energy producing phase, right? Energy requiring phase refers to using up energy. Energy producing phase is exactly what it sounds like. We're now going to start making energy, okay? Now this is the part of the glycolysis that gets a little bit complicated, so bear with me, all right? So going down to our fructose 1,6-bisphosphate. What happens here is something that we call the triose, because it's basically a 3-way reversible chemical reaction. So we have fructose 1,6-bisphosphate can be converted into, reversibly converted into DHAP, as well as glyceraldehyde 3-phosphate. Some people call this G3P. The important thing to note about both of these molecules, DHAP and G3P, is both of these are 3-carbon molecules. Going all the way back up here, I know I drew our little glucose guy as a diamond, but really remember glucose is a 6-carbon molecule, right? So it's 1, 2, 3, 4, 5, 6. So once we get down here, we have effectively cut this molecule in half, thanks to an enzyme called aldolase. So we have 2 3-carbon molecules, and each one of these goes through a series of steps. Basically what happens, let's start with glyceraldehyde, because I think that'll make things a little bit easier. So starting with glyceraldehyde, we have a reversible reaction. So it's driven by the concentrations of each molecule on either side of the reaction in the cell. We have a type of enzyme called glyceraldehyde 3P dehydrogenase. The important part of that name, it is a dehydrogenase enzyme, all right? G3P dehydrogenase. So knowing about dehydrogenases are important because these enzymes catalyze the oxidation of a substrate through the transfer of hydrogen to an acceptor. So let's see what this looks like for us. In this case, we are dealing with NAD+, and then dehydrogenase, G3P dehydrogenase, will basically snatch a hydrogen and donate it to this acceptor molecule NAD+, forming NADH, all right, which is a source of energy, and producing from this, it converts glyceraldehyde 3P into 1,3-bisphosphoglycerate. From here, phosphoglycerate kinase, right? So we're dealing with another kinase and another reversible reaction. Phosphoglycerate kinase does what kinases do best, but this time, instead of stealing phosphate group from ATP, it steals the phosphate group from 1,3-bisphosphoglycerate. So now the kinase is acting to convert ADP into ATP. Does that make sense? So it stole the phosphate instead of from ATP, it stole it from 1,3-bisphosphoglycerate, converting ADP into ATP, right? So that's another source of energy. I'll go ahead and put little things so we can tell which guys make energy. Sound good? This process then converts 1,3-bisphosphoglycerate to now being 3-phosphoglycerate. If we want to break down where this name came from, basically the kinase here stole one of the phosphate groups on that first carbon there and then gave it to ADP, making that ATP, right? So now it's not a biphosphoglycerate because bi refers to 2, it's just phosphoglycerate. And that phosphate group is on the third carbon of that molecule. Sound good? Okay. From here, 3-phosphoglycerate is then converted into PEP via an enzyme called mutase. And then we have our final step in glycolysis for this half of the triose, where PEP is converted into pyruvate, which is, again, a 3-carbon molecule. And this is important because this is a unidirectional reaction. So once we get to PEP, if pyruvate kinase, all right, so another kinase, if pyruvate kinase interacts with PEP, it will convert it into pyruvate, and that will only go in one direction. It can't be converted back into PEP. So just like before, this kinase will also be taking a phosphate group from the substrate here to convert ADP into ATP. More energy. Big yays. All right? So that is this half of the pathway past the triose in glycolysis. Now, what happens over here? Let's talk about it. Essentially what happens here is DHAP can actually be converted into glyceraldehyde 3P or G3P. So basically everything, pardon, that's my blind cat. He'll be okay though, I have him right next to me. Anyways, DHAP can be converted into glyceraldehyde 3P. And this is actually also a reversible reaction. So let me go ahead and fix that really fast. So the arrow is double-sided here. So basically everything that we saw happen over here, it happens over here as well. So basically for every 1 molecule of glucose that goes into this system, we end up with 2 pyruvate molecules, which are 2 3-carbon molecules. And we also end up with 2 ATP molecules. And we also added into the system an NADH molecule. And that's just for this side, right? The same thing happens over here as well, right? Whoop. So that actually means we have a total of 4 ATP and 2 NADHs by the end of this part of the process. Does that make sense? So that is glycolysis, and again, this all takes place in the cytosol of a cell. Now that we have our pyruvate molecule, there are a couple of options, all right?